Hi everyone, it's Mr. Stratton. Today I want to talk to you about nucleic acids. You'll notice the molecule to the side there, which is very recognizable. I'm sure everybody can recognize DNA, which is the main nucleic acid we're going to be talking about today. Now, again, we're going to be focusing on the relationship between the structure and the function of these molecules, and then we're going to compare the structure of DNA and RNA. The functions we're talking about then, storing and transmitting genetic information, and cell energy as well, because ATP is a nucleic acid. The monomer is a nucleotide, and many monomers together make up the actual nucleic acid, so DNA and RNA. And then ATP is actually a single nucleotide. So a nucleotide itself has actually three parts to it. It has a sugar, a phosphate, and a base. So the sugar is a 5-carbon sugar, or pentose sugar, and the two we're worried about are ribose and deoxyribose. So the difference being that deoxyribose is actually missing an oxygen atom, and it's in the name, deoxy. There's a phosphate group, and then one of 5 nitrogenous bases. So 4 of those are found in DNA, and then 3 of the DNA ones are found in RNA, along with uracil in place of thymine. So there's 2 types, the different shapes, the pyrimidines, which are single rings, that'd be thymine, cytosine, and uracil, and the purines, which are double rings, adenine and guanine. Now, how do you remember which is which? Well, one of the ways is, and it's kind of silly, but there's a Y in pyrimidine, also in thymine and cytosine, but additionally, they're whiny molecules. They're really whiny because they say, why do I only get to have one ring? They're small, little whiny bases. Maybe that helps you. Maybe it doesn't. Whatever. But, and then uracil is actually very structurally similar to thymine. But, all right. The structure of DNA itself is a double-stranded double helix, and the backbone is made up of sugars and phosphates. So the sugars and the phosphates form the blue ribbon in the diagram to the side there, but down below you can see that it's actually the sugar and the phosphate, and they're linked together, and then the other side has that as well. What makes up those rungs in the middle then would be the nitrogenous bases, so the A's, the T's, G's, and C's. Okay, they're complementary. The 2 strands complement each other because they match up. A with T, C with G. You guys probably learned that in freshman biology. I don't remember how you remember which pairs with which. Apple with tree, cat, or chips and guacamole, or cat, gato, aunt, tia. However, some way for you to remember. But it's always a purine with a pyrimidine. And they actually figured that out early on when they were looking at the structure of DNA, because they figured out that the percentages always matched up, plus they knew that the size of the DNA molecule, it needed 3 rings across. So 1 double-ringed purine and 1 single-ringed pyrimidine. Okay, so A and T, they actually form with 2 hydrogen bonds, and then C and G form with 3 hydrogen bonds between them. That's the dotted lines in the diagram at the bottom there. Now, one thing about that is that if DNA has more C's and G's in it, it's actually stronger. Like the 2 strands hold together stronger because there are more hydrogen bonds holding the strands together. It's also anti-parallel. Now, what does that mean, right? Parallel means 2 things that run alongside each other. Well, in this case, they run alongside each other, but in opposite directions. And those directions, we know them as 5' and 3'. So you'll notice that the molecules here in the diagram actually have, and let me go ahead and draw on this. The molecules have these little numbers, right? And those are actually the carbon atoms within the sugar. And this end up here is the 5' end, because the 5' carbon is facing up here. And this end is the 3' end, because the 3' carbon on this deoxyribose is facing down. You'll notice the other strand is the exact opposite, runs the opposite direction. 5' is down here, 3' is up here, and so there's your 3' carbon, and down here is your 5' carbon. Now that's going to be important later on when we start talking about DNA replication, because the DNA enzyme, the enzyme that builds DNA, DNA polymerase, only builds in one direction. Think of it as like the 5 freeway, where we've got the 2 lanes running in opposite directions, and then when you want to build redo the freeway, the machine that actually builds the freeway only has a forward gear. Then you're only going to be able to work in opposite directions. You can't go backwards. Function-wise, storing and transmitting genetic information. Right? You've inherited your DNA from your parents, 50% from each, and the DNA is crammed into the nucleus of your cells. There's actually about 6 feet of DNA inside of each cell, so it's really tightly coiled up. It's wrapped around these proteins called histones, which are then coiled into chromosomes. Structure of RNA is slightly different than DNA. So RNA typically is single-stranded. I say typically because there are exceptions. For example, the coronavirus actually has double-stranded RNA. So does HIV. The sugar phosphate backbone, it's basically the same, except the sugar is different. In this case, it's ribose instead of deoxyribose. And again, as I said earlier, the difference is there's an oxygen here in this position, whereas DNA is missing that oxygen. It's deoxyribose. It's missing an oxygen. The other difference is that thymine has been replaced with uracil. Now, thymine and uracil are very, very similar. The only difference between them is that this methyl group right here in place of a hydrogen. And in fact, the other name for thymine is methyl uracil. Okay, the function of RNA is actually in some ways similar to DNA. It can also be genetic information. Some viruses only have RNA, and that is their genetic information. And in fact, that's one of the pieces of evidence that show that RNA actually came first. But in our bodies, primarily RNA is used as the blueprint, the cheap copy of DNA, the blueprint for building proteins. So this is the coronavirus, and those proteins on the outside, the glycoprotein there, the pink protein there, is actually coded for by the RNA inside. All right, the final nucleotide we're going to talk about here is actually ATP. So ATP has a structure of, it's got ribose as its sugar. You can see that it has the oxygen atom right here. That makes it ribose. Okay, this is actually adenine, or A, and then it's got 3 phosphate groups. So these phosphate groups here are basically how it stores the energy. And so ATP, adenosine triphosphate, has 3 of them. If you were to take one off, and that would give us some energy, then it would be ADP, adenosine diphosphate. Take off another one, it becomes AMP, or adenosine monophosphate. Function-wise, ATP is there for energy transfer. So like I said, take off a phosphate group, and you've got some energy that gets released. So to form the bond, it's going to require energy, and so it's how our body stores energy. Something called the ATP cycle, where we eat food, form the bond in ATP that stores the energy, and then later on when we need it back out, we break that bond and get the energy back out for use to do cellular functions. All right, what I want you guys to do now is I want you to identify the 3 main components on this nucleotide. So what are the 3 things, and try to be as specific as you can. Predict whether this would be in DNA or RNA and justify your prediction. And then, is the nucleotide base a pyrimidine or purine? Explain how you know. Complete this table comparing DNA to RNA, and then describe what we mean when we say DNA is anti-parallel. Finally, how is ATP used for energy storage? Thanks for watching.